Okay, it's 2 o'clock. So welcome back to the third session in this series on scaling AI. I'm really glad to see so many people back. We've had 2 days of really interesting, exciting talks already, and I'm pretty confident that the third day will not disappoint either. So thank you very much. I also wanted to, before we start, thank all the many people who are involved in this session. It was initially 2 sessions. We had so much interest that we went over 3 days. And I know that several of the people who are on this list of names are here today. Perhaps you'd like to wave so that everybody can see you. Yeah. Hello. And thank you also to Rukaya and to Kit, who couldn't be here for this session, but did a lot of work behind the scenes with the checking and making sure that all the talks were of sufficient quality and so on. So I think that's everything from me. So our first speaker today is Michael Elliott from the University of Florida. Yes, please come on up.
Hey everyone, I'm Michael at the University of Florida with the ACES lab and with IDIG Bio. Is this the, sorry, I should have looked at stuff. Where do I point it? Over here? Okay. Other way. Here we go. I see. Thank you. Okay, ooh. Okay, so yeah, it's the, sorry, we're getting there. Okay, I'm going to be talking about AI-assisted exploration, curation, and extension of biodiversity data using something called iChatBio. iChatBio is a agentic and conversational interface for biodiversity. You can ask it requests just like you would ChatGPT, so you can form it however you like. The difference being it's an agentic system, so it's based on a whole suite of autonomous agents that are able to go and get data for you that relate to your request and able to process and summarize it and integrate it in a way that is specific to your request. So on that note, it's a biodiversity data discovery and exploration tool. It integrates access to the services and data providers on the right side of the screen, like BHL, GenBank, IDIGBio, GBIF, iNaturalist, and Globi. And actually, just this morning, we added OBIS and ALA. It's a community-driven research platform. I'll share more on that later. And one thing I should note is it's still very much a prototype. It does some things pretty well, but if you go outside of those bounds and ask it hard things that aren't super easy to derive from the data, it might have some trouble. So how does it compare to chatbots like ChatGPT? What makes it different? I asked this question, where can I find Cattleya trianae to ChatGPT? And here's its response. It says the distribution according to major data sources. And to clarify, I specifically asked it search GBIF, IDIGBio, and iNaturalist because I wanted it to be based on data. And its response says on GBIF, the species is listed under taxon key. There's the key. And then it says it's accepted as a species. It also sneaks in a little bit from Plants of the World Online that says it's native to Colombia. It says for iNaturalist that the species page indicates it is endemic to Colombia, which is expected. And then for IDIGBio, it actually wasn't able to do anything, and it just offers the user should probably either use the IDIGBio portal or the API. And then I asked the same question to iChatBio, and it went and it retrieved data from GBIF, IDIGBio, iNaturalist. And then it said, based on the extracted data, the first point, the vast majority of Cattleya, trianae records are from Colombia, and it mentions a bunch of specific regions that are mentioned in the data. And then it also summarizes some of that locality information in the records. It says they often reference towns and rural areas. For the next point, it says that some of the records are in botanical gardens outside of Colombia, like the United States and England, which makes sense. And there's also a handful of iNaturalist observations that list unexpected locations like Australia and Indonesia. And it understands that this is probably because of either cultivated plants or misidentifications. And if you want to go back and see where iChatBio got this information from, on the right side of the website, you'll see links, these clickable buttons that link back to the actual data that was retrieved from the APIs. So now that we know that the species is endemic to Colombia, I asked it, find literature about Cattleya trianae in Colombia. And this is sort of a pattern that it usually follows or is instructed to follow in its responses. First, it repeats its understanding of your request. So even if your request isn't super clear, it'll say what it interpreted your request to be. So it says, I understand you're looking for literature about this species. And then it says what it's going to do. So it says, I'm going to search BHL for relevant articles and publications. And then it adds the disclosure that sometimes it makes mistakes, sometimes it doesn't get all of the data, sometimes the data themselves are not complete just at a global scale. So anything that it says should be reviewed and verified against the data, either that it finds or other data. When iChatBio actually interacts with a data provider or data itself, it'll pop up this little box. So here it says searching BHL. It says it found something, and then it looks at that thing, which is generally just called an artifact. So in this case, it would be an API response from BHL. And it says it found a couple of different articles or journals. I'm not exactly sure. So if I click on that searching BHL box, we can actually see what it did. This is a very, very simple case. It just generated search parameters for the BHL API, and then it submitted a GET request. So it formed a URL to the data. The reason we don't share the URL here, though, is because BHL requires an API key to use it, so we can't share that URL here. Now, if we go back and we look at the response, it has these view here buttons. So it actually hyperlinks back to BHL. So it always points the user back to the source. Now we ask, can you find the accession number of the type specimen of Cortinarius coldingensis, or something like that? And it begins by searching iDigBio. It actually doesn't find anything, so it moves on and it tries GBIF. And then with GBIF, it finds something. Unfortunately, we were actually interested in the DNA accession number or sequence number, but it interpreted our request as catalog number. It happens, but it did find something. And then we said, find the sequence record and show it here, because that catalog number is still possibly enough to get it. So it searches the NCBI nucleotide database, or basically GenBank, and it finds a record, and there it is. You can see it's got the species we were interested in. But you'd probably want to check and make sure it's exactly what you were looking for. Whoa. Sorry about that. The next example, iChatBio can actually show and visualize data on maps. So this example is Amaspiza caracuda seasonal migration. So we wanted to know where the species is at different times in the year. We asked search GBIF for 5,000 occurrences of the species since the year 2000, show them on a map and color them by month. So iChatBio goes to GBIF, it downloads those records and it processes them, and then it builds a map. And then this is the map that iChatBio generated. And you can see in the bottom, the months are on kind of a heat scale, which is a little weird, but basically blue means earlier months, red means later months. So in the south, we see blue and red, which are the winter months, right? December through February. And then in the north, we see these yellow dots, which are in the middle months, which summer. And then in between those, we see little pockets of greener colors, which are spring. And if you're interested, sorry, in how iChatBio actually produced it, you can click on the little boxes that pop up and it'll say exactly how it transformed and queried the data. So this is a query language for JSON, which is what most of the APIs return. So iChatBio generated this and performed the processing. So you can make sure it's showing exactly what you wanted. Another quick example, we asked, are Asian hornets moving north in Europe over the last 20 years, and searched GBIF, and we had a little back and forth, and then asked map only the 2 northernmost points for a year. The idea being the northernmost extent of the species. We expected it to be drifting north over time as an invasive species, and that's exactly what we see here. The blue colors, the colder colors, are earlier years going back to 2005, and the red colors are going to 2025. And you can see on the map, there's a clear trend from blue to red, from south to north, from France to Sweden. Now, getting into a little bit about how iChatBio actually works. It is a multi-agent AI, as I mentioned in the first slide. So what you see on the website is just a front-end interface that lets you enter these questions. So the example question, where can I find Cattleya trianae? That goes to what's called a chat agent. And this is kind of like the front desk for the system. It processes your request. It decides what do you need and which services can I use to fulfill this request or find data related to the request. And those services are encapsulated in expert agents, which are completely separate from the chat agent. They're very narrowly focused, and they're designed to interact with a very specific use case or data provider. Here we have GenBank, BHL, INAT, GBIF, and iDigBio. And like I said, we just added ALA and OBIS. And there's also an agent just for processing data and creating those maps. And so a chat agent by itself, just using an LLM, can still make what's called hallucinations, which are when it makes up information that isn't necessarily faithful to the data and probably not true. Is that a time? Oh, okay. 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 Okay, sorry. Yeah, so we need these supervised agents to rein in the AI and make sure it's not making mistakes. That's not there yet. That's a feature for next year. So maybe next Living Data or Tadwig or whatever it's called. We also accept agents from the community, new agents from you. We also have a bunch of agents being developed by students right now. These agents are developed using something called the iChatBio SDK, which is a Python package. It's available on the Python package index, just like any other package. There's a QR code there. And it's the whole point of this SDK is to make it super simple to develop agents. You can see this is a hello world example. It's only about 30 lines. The left side is just saying what your agent is, and then the right side is only about 10 lines of logic. And all of this is enough to deploy a web server that can communicate with iChatBio. So it really streamlined development. Sorry, I'll skip through. But the iChatBio, it allows multilingual capabilities on top of these agents, so your agents don't need to worry about multilingual abilities. It also provides vision capabilities, so your agents can provide images and iChatBio can work with them. Yeah, the whole point is that iChatBio handles a bunch of stuff so that the experts that the community develops and we develop can be very narrowly defined and implement exactly what it is that you're interested in. And that is all. Read those bullet points. That's what, is important. And I just want to acknowledge all the students who helped us work on this and develop iChatBio. Thank you all.
Yes, thank you very much, Michael. I'm sorry to cut you off there. Our next talk will be an online talk from Christian Billing. And just as a reminder, we are holding all questions until the end. So please stick around. There'll be time for discussion after all of the talks. Thank you very much.
Thank you for the opportunity to present the KeyBids project that implements a framework for information extraction from natural history collection specimen labels, and potentially also from other written sources. We estimate that the collections at the Museum of Natural History Berlin comprise more than 30 million collection objects. We don't know exactly. Some parts of the collections have not been touched for a long time, and the process of inquiry into the contents of our collections and the publication of all acquired data using digital formats is an ongoing, long-term process. With these millions of objects also comes a wide range of associated written records, inventories, scientific reports and notes, travel and expedition reports, sketches, drawings, and diaries, to name a few. Specimen labels are one type of object-related document that is especially relevant for collection management and collection-based research. Usually, each collection object is accompanied by one or more labels on which the basic information about the origin of the object and its provenance have been recorded. This data is crucial for a number of collection-based research fields. Among others, The labels provide data about past occurrences and abundance of species in the past, providing baseline data for analyzing temporal changes in biodiversity. Mobilization of specimen data from the collection labels is part of the long-term project collection discovery and development at the museum, which integrates the process of handling and updating the collection infrastructure, ensuring proper conservation, transferring collection object data into the digital realm, and making that information accessible to different stakeholder groups. Within the context of collection discovery and development, mobilization of collection label data is a three-part process. The first step being digitisation of collection labels. The second step is to extract the information that is encoded in the digital images of the original paper documents. This is what the Curation project is about specifically. The third step is preparing the extracted data for sharing in versatile and standardized data formats for downstream use, using, for example, semantic web technologies. The task of information extraction from digital images of the collection labels can be subdivided in 5 functional steps. With the digital image acquired as a starting point, the first step is to pre-process the image for optimum results in the subsequent image analysis steps. Layout recognition, as a second step, identifies image regions which actually contain information of interest. In the case of collection labels, these first and foremost, the image regions that actually depict labels and the lines of text within them. The third step is the actual text recognition, that is, to recognize individual symbols on a syntactic level. Semantic parsing then tries to classify parts of the text denoting, for example, institutions, identifiers, tax as your persons, entity disambiguation, and linking as a fifth and final step, attempts to identify individual entities denoted in the text by linking them to identifiers in authority, record repositories like Wikidata or the VIAF resource for persons, and the Catalogue of Life for taxon names. Let's look at the inputs and outputs of the Curation data pipeline. What enters the application and what comes out. The input are the document images plus configuration parameters for the pipeline. The workflow output for a given image is a page XML file with image annotations. 1 page XML file per input image. Optionally, intermediate data of the pipeline run can be requested. If the pipeline is run in evaluation mode, evaluation metrics from comparison to reference data is provided. Let's go through the individual steps of the pipeline. The purpose of image preprocessing is to prepare the image as optimal input for layout analysis and optical character recognition. The input is the original image and the output of this step is a processed image. Currently, we have implemented a pre-processing module based on OpenCV with steps for resizing the image, for grayscale conversion, for visual noise reduction, for contrast enhancement, and for image binarization. The layout analysis step identifies image regions which actually depict labels. The input is the processed image from the previous step, and the output are the coordinates for bounding boxes of the regions of interest. In the current release, this module is based on the Segment Anything framework. For the character recognition step, the input is the processed image and the bounding box coordinates. The output is a transcript for each bounding box. Curation currently implements 2 different modules for optical character recognition. Firstly, EasyOCR as a tried and tested solution for optical character recognition. Secondly, with Moondream as an open source and locally deployable framework, visual language modeling technology is leveraged for Curation. Moondream is parameterized with a prompt. The default prompt, which can be changed as input, parameter reads as the following. Transcribe all printed and handwritten text on this label of a specimen from a collection of a museum for natural history, being especially careful to preserve any scientific names, dates, and location information. Maintain the original formatting and line breaks. Most text is in German. Semantic parsing of the transcript aims to identify parts of text that denote object classes of interest. For example, dates, IDs, persons, or places. The output of this step are character sequences from the transcript tagged with class signifiers. Currently, we have implemented a module based on SpaceI and regular expressions passing for dates, URIs, biological sex symbols, geographical coordinates, and type specimen signifiers. We have determined that a rule-based approach based on regular expressions is more efficient than a statistical model for these categories, as we deal with clearly definable syntactic patterns for these categories and are faced with context-sparse documents. Entity linking as the final step of the information extraction pipeline aims to identify named entities in the transcript with authority records. Its input is the transcript with all annotations from the semantic parsing step. Currently, entity linking in KaiBids is implemented for geographical place names through the GeoNames API. Let me highlight some of the challenges for information extraction at this point. With Easy OCR and Moondream, we have implemented two capable solutions for typed text. The performance for handwritten text is poorer. This is one area where we hope to implement more capable modules in the future. Another challenge is detection of non-script symbols, which also carry important meaning. For example, a red dot signifies type specimens in a number of our collections. For strike-throughs or additional comments, we still have to figure out a good way to recognize those and then to extract and represent the relations of these parts of the transcript. A fourth issue is identifying a reading order on the labels when labels have multiple text parts arranged in irregular ways. For executing information extraction workflows, there are 3 layers in the KeyBeads application. Individual workflow runs are defined by a workflow template. Workflow templates are deployed and orchestrated by a controlling outer layer. KeyBids uses Prefect as a workflow orchestration tool to schedule, monitor, and manage workflows. On a practical level, Prefect provides a graphical user interface for initiating workflow runs and for accessing workflow data. In conclusion, the KeyBids framework provides a structure for implementing data pipelines for information extraction from specimen labels. It's open source and locally deployable with reasonable hardware requirements. The source code is available on GitHub, and we're happy for anyone to try it out or fork it to open an issue or a discussion. We look forward to interacting with a larger interest group for label information extraction. In implementing the KaiBids application, we tried to not reinvent the wheel, rather to combine existing state-of-the-art frameworks. An important design goal for KeyBids was adaptability for newly emerging technologies, whether these can be deployed locally or over the cloud, and for alternative implementation of individual information extraction functions. Modularity and adaptability also are important for our approach to start, with default configurations of each component to adaptations for specific use cases if needed. What's the outlook on the further development of the application? Certainly, we can add to the user experience for more visual support and for improved Prefect integration. We want and need to improve performance for handwritten sources, and we have some very exciting developments in some other projects that we hope to be able to integrate in time as updated workflows into the KeyBids framework. We'd like to broaden approaches for semantic analysis, for example with relation analysis for context-sparse documents. I would like to acknowledge the entire project team and the support of the AI Eco Lab, an initiative of the German Federal Ministry for the Environment, Climate Action, Nature Conservation, and Nuclear Safety. Thank you for your attention.
Thank you very much, Christian. And next we will have Elamina Mimouni from the Quebec Centre for Biodiversity Science. Please take it away.
All right, so just to make sure. All right, so hello everyone. Today I'm going to talk to you about what we have been doing at the UCBS and Biodiversity Quebec. So I'm going to talk about biodiversity data sets as linked and open data in research and conservation. So throughout this symposium, a theme that has come up a lot of times is the concept of fair data. By enabling fair data, we allow for transparent and open science, standardized collaboration, and robust data stewardship. But how is this achieved? How can we implement it is another issue. How we can do this using linked open data shall be shown using a test example, which is the Broke West fish dataset, a dataset that consists of a vessel that surveyed the Antarctic coast for fish communities, and that has been recently been republished onto the GBIF test IPT. This data set is actually a complex data set because it consists of not only occurrences and events, but also additional entities such as media, pictures of the fish that were caught, material entities, the fish themselves, the specimens, where are they hosted, assertions, measurements that were made on these material entities, as well as the entire sampling program that needs to be transcribed and integrated into the data set. And if you were here on the first day, there was a panel or a session on the Darwin Core data package that discussed how this data set done by Yiming Gang can be transcribed onto the recently proposed Darwin Core data package using the recently proposed Darwin Core data package terms. In our case, we propose that in addition to simply the relational database approach, this data set can also be represented as a directional graph using RDF and semantic web technologies. What this means is that the entirety of the 16 or 17 relational tables can all be represented as a single directional graph, wherein every entity is a node, a typified node. So you'd have nodes that represent assertions, node that represents events, node that represents occurrences, and so forth. And that all these entities are related through semantically meaningful terms. So since these are all typified nodes, every segment in this graph represents a OWL object property. So this graph would be readable as you would read a semantic phrase. So you could say that an assertion is made on a material entity that was captured on an occurrence that was captured on an event. So it is semantically sound, and the terms are based on the data package terms. So it uses Darwin Core as an ontology. And in addition, since we are using semantic web technologies, these nodes are not simply connected with each other, but also to resources on the World Wide Web. By that, we mean that every node is given a URI to make it findable and identifiable. And if you were to consider, for example, an agent, someone that carried out the sampling, instead of a UUID or an ID, you would have the ORCID page of the researcher that made that sampling. If you were to consider a media, you would have as a URI the Zenodo page of that image. If you were to consider a bibliographic resource, such as what item was taken to identify the specimens, you would fall upon the Internet Archive page for said book, and so forth. So this makes sure that every resource is findable and connected with the resources outside on the World Wide Web. And one other thing is that in order to interact with these graphs and to query them, you get to use Sparkle, which is an awesome query language that allows you to extract parts of the data that you want. So for example, you would have a query here, a Sparkle query, that will allow you to query for any data set that has a material entity of Pleurogramma antarcticum, and optionally, you can ask it for a measurement or fact. So any measurement, if available, please give it. And you can see here that every term is prefixed by Darwin Core or Darwin Core IRI prefixes. So we are using Darwin Core as an ontology, but we are also considering other ontologies such as Dublin Core and EML, so ecological metadata language. And Sparkle is also very important for one additional feature, but for several additional features as well, due to the fact that Sparkle can consider federated queries. By federated queries, we mean a query that has a service clause in its query. And what this does is that it allows Sparkle to not only query one endpoint, but several endpoints. So if you were to take this query, for example, you are requesting the scientific name as well as a set of proteins. But how is this built? How is this query broken up? You would request any occurrence that's a Darwin core occurrence and that has a scientific name and is part of the Broke West fish data set. So essentially you are requesting every species that was recorded during the Broke West data set, but also you are querying the Sparkle endpoint of UniProt, which is a European protein database, for the exact same scientific names, but you are requesting on that other database, so not your database, the database of UniProt, you are asking for any protein whose function contains the term respiratory chain. So considering Sparkle and semantic web technologies truly allows interoperability, as you would expect in these technologies. So you have just made Darwin Core interoperable with UniProt ontologies. And in conclusion, I would just say that if we truly want to achieve open and fair data sets, RDF and semantic web technologies do not just represent an alternative way to represent the data sets, but truly a crucial and essential step if we want to truly achieve linked open data sets. And on that, I would like to thank all my colleagues. Oh, there was supposed to be a thank you note for colleagues. Thank you. Oh, thank you for listening. And if you have any questions, do ask them to me either in person or online. Thank you.
Thank you very much. It's a very interesting talk. Next up, we will have a virtual lightning session from Felipe Miranda Suarez from the University of Sao Paulo.
Hello, everybody. My name is Felipe. I'm a researcher at INRAE in Montpellier, France, and I'm presenting today the work we have been doing with a large language model as a semantic engineering assistant, more specifically ChatGPT. So we will talk about the lessons learned and also the limitations we faced doing this work. So dealing with taxonomic names in ontologies, in controlled vocabularies, it's quite challenging, especially when we have these concepts coming from multiple countries in different languages. So here we have an example of Wikipedia. If we translate the page of pimenta, which refers to these radish berries that come from the genus Capiscum that are commonly traded in Brazil. If we switch the language in Wikipedia from Brazilian Portuguese to English, we will have a completely different page for black pepper, which refers to Piper nigrum, a completely different species. But why does that happen? So the automated translation of pimenta, of this term to English, is frequently done as pepper. And we know that in English, pepper more commonly refers to black pepper. And the word most often used for these radish berries here are chilies, is chilies. So sometimes that's why the systems, they model these two concepts this way. But the way to tackle this down is properly modeling the relationships between the species and the product type, so we avoid this kind of confusion. And besides that, we also have the problem that many ontologies, they deal with these scientific names manually in their vocabularies. So here we have an example coming from Embrapa in Brazil, which a synonym, the name of a fish, which is a synonym, is used as a preferred term in their thesauros. Or if we check, for instance, the JBIF backbone taxonomy, we will see that this is considered a synonym. So this automated approach with the assistance of a large language model, we used to solve those issues, and we tested it to when we were building the agricultural prototypes ontology. So here we have a part of this ontology that it's only for the species names, and then we have another part of this ontology for the product types, and then we were relating both of them to avoid these misunderstandings between the common name and the scientific name. And we followed this pipeline where basically the users select a list of species. This list of species will be sent as a request to the JBIF backbone taxonomy API. And then this algorithm should check, the semantic engineering system, ChatGPT, should check the accuracy of the names in this list. So for instance, if it's a synonym, then it should fetch the accepted name and from there extract the taxonomic classification for that species and finally generate an null ontology based on that extracted data from the API. And for that, we adopted two approaches. The first one using ChatGPT with the browser up plugin. This was done quite a long time ago, like 1.5 years. So the plugins were still active, but now they've been replaced with the GPTs, and we also developed a GPT to do to perform the same task as in this first approach. And then in the second approach, we asked ChatGPT to develop an algorithm that would be able to perform a very similar task. So in the first approach, we ChatGPT, with this connection with the JBIF backbone taxonomy API, successfully managed to detect all the synonyms in the list of species and model it properly in OWL. But without the connection, without the access to the API, we did some tests and some hallucinations appeared. So for instance, in some of the experiments, it said that Euterpe edulis and Euterpe oleracea, which are two different species of palm trees, were the same species. So without the connection to the API, we concluded that the modeling suggestions, modeling recommendations were not reliable. But in this first approach, we had a scalability issue. So the model would fail for very long lists of species. And so we understood that the safe way to go would be providing smaller lists, at most 3 species at a time. But this is also quite lengthy. So the whole process is quite long. So for long lists of species, we recommend using the second approach that I will talk about. And as I told you, we are also finishing the development of a GPT called Taxonomy Overlizer 2.0 that will perform the same tasks. So, and the second approach, as I mentioned, was a Python script that we asked ChatGPT to generate to perform the same task. So we overcame the scalability issue. There is no risks of hallucination since this algorithm is LLM independent, but it's typo, this algorithm is typo sensitive. So in our input data, we have some typos in the scientific names, and the algorithm struggled to provide the serialization for those species names that had typos. And in the end, we also deployed this user interface in this web application, where basically the users, even if they don't know any programming languages, Python and so on, they can just use this interface to provide a list of species and get the OWL ontology out of that. And as final considerations, we understood that LLMs can speed up ontology engineering, but it's still, we need experts to check the outputs and validate and decide if they can be reused in real world ontology implementations or not. And that's it. Thank you very much for your attention.
Thank you, Philippe. Now we have an in-person lightning talk from Ivan Lebras from the Données, France.
Old MacDonald had a farm. Hey, hi, hey, hi, ho. So I'm Ivan Lebras, and I will speak again from Galaxy Ecology, but here on something we are making on AI. I have 16 slides, so I think I just need to say to you to look at the paper describing Galaxy Ecology initiative, and I will go really rapidly. The idea is to share source code so you can reuse it. from the web without having programming knowledge, and you can reuse computational workflows to produce biodiversity metrics. We have a lot of trainings, so don't hesitate to go to Galaxy Ecology trainings and look at that more into detail. So something quite new since the beginning of the years is AI, at least for Galaxy Ecology. And we are first of all working on a training will be published soon, allowing to show people how they can apply YOLO-based models to identify species from pictures on the deep sea, completely from the web without having programming knowledge on a workflow who can be shared and reproduced quite easily. So stay tuned. So to give you some insight why we are working on AI now, in fact, we are working on citizen science projects since years and notably on crowdsourcing. I have no time to go further, but we have tools to annotate data, acoustics data, and now images data, and so on. And the focus I want to produce you is, I don't know how to make beautiful slides, so I made a lot of shitty things, sorry. So I want to illustrate in 3 use cases we applied, just trying to see if we can reuse the Galaxy platform to apply to ecological questions thanks to AI, but again, without coding, on the web, on a collaborative environment. So the first one is related to creating deep learning models completely from Galaxy. Another one to use interactive annotation and train and label. And the final one is something I was testing just 2 weeks ago, bless you, to go from camera traps and just take models created by your colleagues and share the models and a workflow who allows people to upload their video so they can identify the species on the model without having programming knowledge on the internet and things like that. And so I made some beautiful screenshots just to see you the reality of the things when we are on Galaxy. And for example, we want to apply a model. Everything is tooled into Galaxy. And for example, there you have a series of tools who allows you to create a deep learning model architecture, then continue to modify the model, train the model, and evaluate this. And what you see is that each time, each step, you have a complete traceability of the provenance on what we call the Galaxy history on the things you are analyzing. Then you can evaluate your model. Then you can make prediction. And at the end, you can categorize, convert to a class vectors, and see a beautiful matrix. So this is maybe quite trivial for people making AI, but this is doing only on the web, completely open to anyone. And we successfully reproduce the figure who is produced on the paper. It's not often we can reproduce things from paper. Another thing is related to annotation of images to train labeling. So here the workflow is to use an interactive interface where we can labeling, for example, the shrimps or crabs, and we can use AI model to help modeling, make the annotation too. And after that, we have tools to convert the annotation, to perform training, and to make image labeling. And the final one is how we can use Jupyter notebooks, something like RStudio for those who didn't know, to capture a code who is made by a researcher and the model, so we can, in 1 day or 2 days, push it into Galaxy, create a workflow, and share it with anyone who want to use it, just clicking on a button after having uploading a video file. And this look like that. Last news is that we have connection from Galaxy with Hugging Face models who will be soon merged. And I want to thank you for your attention and all the team of young guys who helped me since 8 years to just contribute to this open community around Galaxy and Galaxy Ecology there. Thank you.
Yes, thank you very much, Ivan. Our final talk of the session will be a virtual talk from Regine Leb.
Since the 1980s, artificial intelligence has been used as a decision-making tool in areas such as medical diagnosis and biological identification, first through what was named expert systems and later through deep learning. Today, we propose to combine symbolic and data-driven AI in hybrid scenarios for identification, while keeping interpretable reasoning. We distinguish two kinds of AI depending on how they represent and process information. Symbolic AI expresses knowledge as logical rules in knowledge base. It provides explainable logical reasoning, but requires expert work to encode that knowledge. Statistical or connectionist AI, on the other hand, learns from large datasets. It is data-driven. Neural networks like CNN are powerful, but often considered black boxes because their internal representation are hard to interpret. Why hybridation? We already have a validated system based on knowledge bases, expert-free, and we aim to combine it with deep learning to benefit of connectionist AI efficiency, reduce errors, speed up the identification process, and preserve explainability of the results. For that, we designed 3 scenarios where deep learning is integrated at different steps of the identification process. Before the decision tree to reduce and target the knowledge base, during the use of the key to check user inputs, throughout in a dynamic interaction between both AI. In all cases, AI models are trained to classify taxa or traits, while expert knowledge base provides a logical framework. In the first scenario, deep learning is used to target decision tree. For instance, a CNN can be trained to recognize the orientation of an insect on a photo, dorsal, ventral, or lateral view, so only the visible characters are then used in the key. In another example, a CNN is trained to classify bees by morphotype, and the identification key is then restricted to the corresponding species. With this scenario makes identification faster, more accurate, and context sensitive. In the second scenario, deep learning verifies user choices during identification. If the CNN detects a mismatch between the photo and the selected state, the system sends a warning and suggests a recheck. In educational context, this helps users interpret terms correctly. The third scenario involves a continuous interaction between symbolic and statistical AI. In coral reef monitoring, for example, video transects are processed into images, coral are detected on the images, we use YOLO, and then identified. For identification, the knowledge base creates a dynamic key by selecting the most discriminant information to distinguish the remaining taxa. Depending on the results, the system chooses the most suitable tool, symbolic reasoning or statistical recognition for the next step. The process continues until to obtain the identification result. This dynamic process evolves over time as the knowledge base or classifier are updated. To conclude, deep learning is becoming an increasingly accessible tool for automating identification. Yet, knowledge-based systems remain essential for interpretability. Symbolic reasoning also has a lower environmental impact than data-intensive methods. We are now developing a new version of our software where this hybrid scenario will be tested. Thank you for your attention.
All right, it's a very interesting set of talks. We have some time now for questions and discussion. So could I please invite all the in-person speakers to come up on the stage? And if you have questions, put your hand up, please. Wouter will bring you the microphone, and yeah, then we can make sure the online people can hear you as well. So could I invite all the speakers up on the stage again, please? Did we only have two?
No, thought there was
Yeah,
three.
Yes, so we have three in-person speakers today. Do we have any questions for any of these three talks? Yes, Andrea. Microphone is coming.
Hello. Well, thank you to all three speakers. I have a question for Michael. So how did you pick and choose your agents and which ones worked best? And what did it work best with mentioning all three agents in your prompts as well?
Well, as far as picking them goes, well, we started with IDIC Bio because, you know, we are IDIC Bio. And then GBIF seemed like a high value, low hanging fruit, very similar to IDIC Bio in terms of interface and types of things. But really, it was just we compiled a big list of data providers and sort of sorted them by what seems to have the most impact and fits the best with the other agents we already had, and just went down the list. And we're still going down that list, and we probably will for a while.
Yeah, thank you.
Yeah, question from Klaus at the back there. Just the microphone is coming.
Yeah, thanks to all speakers for these talks. That's the question for Amin. I have a question regarding your federated query. Have you tested how this scales? Because if I got your code right, you query the full graph of Uniprot, which is probably one of the largest bio or omics graphs anyway.
Yes.
And then you do a filter afterwards. So you have a full graph retrieved and filter for it. Does it scale if you do more of these service queries?
Well, I've experimented different ways. Yes, you can use it, but it's true that with Uniprot, there are some scaling issues. But what I find is that if you use Apache Fuseki as the server and use instead of a regex, filtering regex, if you use Lucene to index all the text terms, then it is still doable. But if it's a really, really big query, then yes, you'd be better off doing it by chunks. So yeah, there are different strategies depending on the size of your query. It's mostly trial and error.
Thank you. Do we have any more questions? Is there a question at the front? Yeah.
Yeah, I've got a question for you, Ivan. Then that would be, is Galaxy in some way easy to connect, like when you train the model, is it easy to connect with something like Hugging Face and like getting back and forth with your, or are you on your own repository of models?
That's I tried to say, but the breaking news is that there is a pull request on the Galaxy project to allow the direct connection with Hugging Face. So it will be soon available on all the Galaxy environments, so Galaxy US, Europe, Australia, on your own, and Galaxy Ecology notably.
That will go both ways, right? So, yeah, cool.
Sorry, just to follow up. So this means you can run everything locally as well then, using your pipeline. So it's possible to run everything locally using your pipeline as well?
Yes, it is not the way we prefer that people are using Galaxy, because it is really to put on HPC with high resources and shared resources. But in fact, you have the possibility to have a Docker instance locally, notably, or on a regional instance. You can deploy quite easily just with the tool you want, just execute the workflow you want, and you can shut down the Docker and stop that. So this is possible. It's not the way I think it's the best to use. In the meantime, maybe it can allow some particular usage on that. Yeah.
Thank you very much. Oh, I see Wouter has a question as well.
Yeah, a question for Michael, if I may. You said that you have a multilingual workflow, and as I understood it, that's a model on top that handles the language. Is it,
Well,
or all the...
It just comes from the fact that we're using a large foundation model. We're using models like GPT 4.1, and they just have a lot of languages built in. But the agents don't need to operate with those kinds of models. They can work with really conventional NLP workflows, they can just print straight up English text or whatever language. And then GPT 4.1 will do the interpretations.
So it's a translation. Will you then get the naming problem that Felipe mentioned, where pepper in English would normally be black pepper, stuff like that? Do you see any of that?
Possibly. I'm not sure. I mean, I've never encountered something like, because I only work in English, so I'm not really sure. And I probably wouldn't recognize it if I saw it. But yeah, that's something that we should probably look into. One thing that with iChatBio, what we try to do is to have agents so that we're not working at the common name level, that we just immediately switch to scientific names. And there would be an agent dedicated for that, like a catalog of life or whoever else is, handling that kind of information.
Thanks.
Yes, do we have any more questions from the audience there?
There is a question in the chat.
There's an online question for Michael. How do you plan the expert validation of iChatBio?
Okay, so that's a really broad topic, actually, because each expert, there's a whole world of different things they can do. But right now, the experts are, to some degree, self-validating. There's a lot of different levels you can do it at. Right now, they just make sure if they're using an API, that the parameters they're using for that API are sensible, so that the API will not return an error. But there's also the different agents have different steps where they'll make sure that the outputs are coherent with the inputs, or they'll do multi-layered processing where they check different logical, consistencies at different layers. Yeah, and then as far as doing validation or verification beyond the agents, one way to do that is to just, there's so many different ways you can do it. One example is repeating the actions of the agent and seeing if you can go back from their response to the source data and sort of follow the logic back to the source, just to make sure they're not making anything up along the way. Another way is simply to repeat agents and see if they give you the same results multiple times. There's really an infinite number of possibilities you can do. But there's not really like a one size fits all, There's many different layers you're operating at.
Thank you very much. I think we have time for one more question, if there are any still out there. Otherwise, I'm sure that the speakers will be around during the break as well. I know that just before I let you all go for coffee, Ariana also wanted to make a few short announcements.
Hello. Yes. Yes, thank you. Just a quick one, guys. Just a couple things so we can keep the momentum going. Firstly, there is an AI Tadwig sub-channel there if you want to be part of that. Secondly, if you weren't in the unconference earlier, I think it was mentioned that some folks here are hoping to introduce a new interest group more related to large language models. So do get in touch with some of us. I think there will be a blog around. Someone correct me if I'm wrong, but there will be a blog around coming soon if you're interested in joining that. And lastly, not too related to living data, but similar group. There's some of us organizing a more general AI and robotics for collection group here. And we're hoping to have virtual meetings every other month and present talks and things like that. It was technically introduced as a CTAF ISTC thing, but we are absolutely opening this up for everyone. So it's not limited to a group. So if you are interested in that, please come to me because our first event is in 2 weeks' time. So it's pretty soon. But yeah, keep in touch, everyone. And thank you all for joining. I'll give it back to you guys. Yeah. Oh, sorry, one more announcement. Sorry, if you were in the organization team here, please come to the front at the end of this session so we can do a group photo.
All right, then just to thank all our speakers once again and the organization team as well, of course, here.